Welcome to this lecture on the CRISPR-Cas9 system. Today, we are going to explore one of the most revolutionary breakthroughs in modern biology. We will understand how this microscopic tool acts as molecular scissors, allowing scientists to edit genes with unprecedented precision, and why it is fundamentally transforming the landscape of animal biotechnology and medicine. Now that we have introduced our topic, let us imagine you are reading a massive, multi-volume encyclopedia that contains 3 billion letters. Now, suppose you discover a single spelling mistake that ruins the meaning of an entire chapter. Finding that one wrong letter and fixing it without accidentally erasing the rest of the book sounds like an impossible task. For decades, scientists faced this exact problem when trying to fix broken genes. This is where our topic comes in. The CRISPR-Cas9 system is essentially a biological find and replace tool. It uses a molecular GPS to navigate through the billions of letters in an animal's DNA, locates the exact spot that needs fixing, and uses microscopic scissors to cut it. Building on that intuitive understanding, let us look at the formal definition. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and Cas9 refers to the CRISPR-associated protein 9. Originally, this fascinating system was discovered in bacteria, where it serves as a natural immune defense against invading viruses. When a virus attacks, bacteria capture a piece of its DNA. If the virus returns, the bacteria produce a guide RNA that matches the viral sequence, pairing it with the Cas9 protein to slice the viral DNA apart. In modern animal biotechnology, scientists have brilliantly repurposed this two-part system. By designing a custom guide RNA, we can direct the Cas9 enzyme to cut literally any specific location in an animal's genome. Knowing what this system is, we must now ask why it is so important. Before CRISPR, editing genes in animals was an incredibly tedious and expensive process that relied on older tools like zinc finger nucleases. CRISPR completely revolutionized biotechnology because it is highly affordable, precise, and easy to program by simply changing a short RNA sequence. In the real world, this technology is being used to create genetically modified animals, such as pigs with specialized organs that could one day be safely transplanted into humans. It is also being aggressively explored in clinical trials to cure devastating genetic diseases like sickle cell anemia by directly correcting the mutated DNA in a patient's own cells. To truly appreciate this precision we just discussed, let us look at the basic mathematics of DNA sequences. DNA is made up of 4 chemical bases. We can calculate the exact probability of a specific sequence occurring randomly in a genome using a simple exponential formula. In this formula, P represents the probability of finding a specific sequence purely by random chance. The number 4 represents the 4 possible nucleotide bases. The variable n represents the length of the target sequence, which is typically 20 nucleotides long for a standard guide RNA. Physically, as the length of our guide RNA increases, the chance of it accidentally matching the wrong part of the DNA drops exponentially, making the system highly specific. Before we move on, let us see this formula in action with a concrete example. A scientist designs a standard CRISPR guide RNA with a target sequence length of 20 nucleotides. We want to find the probability of this sequence occurring at any single location by random chance. We substitute the value 20 into our exponential probability formula. We use the base of 1/4 because we assume each of the 4 DNA nucleotides has an equal chance of appearing at any given position. Calculating this gives us a result of approximately 9.09 times 10 to the power of -13. This incredibly tiny number, roughly 1 in a trillion, represents the raw probability of a random match occurring at any single starting position. Now that we have calculated this incredibly small probability for a single spot, we must prove, mathematically, whether this sequence is unique enough across an entire animal. Finding the target sequence is only half the battle. We need to know if it will accidentally match elsewhere. To find out if it will happen in the entire animal, we look at the genome as a whole. We calculate the expected total occurrences, denoted by E. We do this by multiplying our tiny probability by the total number of base pairs in the mammalian genome, which is roughly 3 billion. This multiplication scales our single-site probability up to the scale of the entire living organism. Continuing with our calculation, we arrive at the final expected number of random matches. The multiplication yields approximately 0.0027. This value is far less than 1. Physically, this means that a 20-nucleotide sequence is so highly specific that it is extremely unlikely to appear anywhere else in the entire 3 billion-letter genome by pure chance. If the expected value were greater than 1, we would worry about the Cas9 scissors cutting the wrong genes, known as an off-target effect. This mathematical proof shows exactly why CRISPR-Cas9 is considered a highly precise and safe gene editing tool for animal biotechnology. Having proven the safety of the target sequence, we must now introduce another crucial component. Finding the target sequence is only half the battle. The Cas9 protein also requires a short specific sequence immediately next to the target, called a PAM, which stands for Protospacer Adjacent Motif, in order to bind and cut. For the most commonly used Cas9 protein, the required PAM sequence is NGG, where N can be any nucleotide, followed by 2 guanines. Without this PAM sequence present, the protein simply slides past the target DNA, acting as a crucial safety mechanism. Because N can be anything, and guanine has a 1 in 4 chance, this PAM sequence appears on average once every 16 base pairs in random DNA. Understanding how frequently this PAM sequence appears leads us to a very practical formula. When planning an experiment, researchers need to know how many potential cutting sites exist within a specific gene. We set up a simple linear equation for this. In this formula, NS represents the total number of potential target sites. The variable L represents the total length of the specific gene we want to edit, measured in base pairs. Finally, FP is the constant frequency of the PAM sequence we just discussed, which is 1/16. By multiplying the total length by this frequency, we can estimate the total number of valid binding spots. If the gene length increases, the number of available target sites increases proportionally. Now that we have our formula, let us apply it to a real laboratory scenario. Suppose you want to use the CRISPR-Cas9 system to knock out a disease-causing gene in a laboratory mouse model. The specific gene you are targeting has a total length of 1,200 base pairs. We want to estimate how many potential target sites are available. We substitute the given gene length of 1,200 into the variable L. We also substitute our known PAM frequency of 1/16. Multiplying these together yields a final result of 75.
Practically, this means the scientist has about 75 different locations where the Cas9 protein can potentially bind and make a successful cut, providing plenty of backup options.
Having calculated the mathematics behind the system, let us now visualize exactly how it works. On screen, you are looking at a schematic of the CRISPR-Cas9 mechanism. The large blue structure represents the Cas9 endonuclease enzyme wrapped around the DNA helix. Notice the guide RNA, labeled in orange, pairing perfectly with the 20 nucleotide target sequence on the unwound DNA. Just to the right of that target, you can see the crucial PAM sequence, labeled NGG, which anchors the enzyme. Finally, look at the scissors icon, indicating the exact double-strand break site. This elegant, mathematically precise mechanism is what allows scientists to correct genetic mutations with unprecedented accuracy. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that the CRISPR-Cas9 system is a revolutionary and highly precise gene editing tool, acting as molecular scissors. Second, we saw how it uses a customizable guide RNA to locate specific DNA sequences, and we proved mathematically that the probability of random off-target matches is exponentially small, ensuring safety. Third, we discovered that the Cas9 enzyme strictly requires a PAM sequence to make a cut, providing a crucial safety mechanism and plenty of target options. You now understand the fundamental mechanics of how this technology is driving the future of life-saving therapies and animal biotechnology.